So, not to be creepy, but I actually started hit record a while ago and I could hear what you were saying. And I heard you say, like, oh man, the cytoskeleton, this is going to be so boring. I can't tell you how wrong you are. What we're going to talk about in these next two videos is some of the most interesting stories we might have to offer in the course. I can't wait to share this with you. But I will give the warning, we're getting a little bit late in the course. This is a 300 level course. It's the spring semester, so that means we're leading you into 400 level courses, especially if you're a junior. So the material is going to start to get denser and denser as we wrap this course up. So it's a video. Pause the video when you have to. You can always go back and rewatch certain sections. You can always go back and rewatch the whole thing. You have access to the slides. You can download the slides from the LMS if you need to. And of course, I'm a resource for you in office hours, text, email, whatever you need. So you're not alone, but just be prepared for the material to get a little bit denser. This is going to be the first half of chapter 17, pages 595 to 615. Today, we're going to first start off actually with a little bit of a broad overview on the cytoskeleton in general. And then we'll take about half the video probably to talk about intermediate filaments, the role of intermediate filaments in supporting cellular structure, the anatomy of intermediate filaments and how intermediate filaments are built, the role that intermediate filaments play in the nuclear envelope, and then we'll move on to microtubules for the second half of this lecture. Talk about what polarity is in microtubules. They actually have two different ends, and we'll talk about why. Just like the intermediate filaments, we'll talk about the anatomy and physiology of microtubules, how they grow, how they assemble, how they're structured, and of course, what they do, which in a nutshell is organize the interior of the cells. If you remember some of your cytoskeletal instruction from previous courses, you might notice that actin is missing from this list, and that's because the next video is dedicated entirely to actin. So I truly believe that a lot of what we've been discussing in the last few weeks, especially neuronal impulses and action potentials, and of course cellular signaling and G protein coupled receptors is pretty amazing. But cells do a lot of things that aren't so amazing as well. They maintain and sometimes change their shape. They organize the location of their organelles and other interior compartments. They interact mechanically with other cells in the environment, and they carry out coordinated movements. So, kind of like housekeeping stuff, the daily grind of just being a cell. But they're cells. So, if you think of even this list, this is pretty amazing. What am I talking about? This is amazing too. And when we get into the details, we're going to see just how amazing these processes are. Everything I've said here, shape change, interacting with other cells, interacting with the environment, all of those processes and many, many more are mediated by the cytoskeleton. The cytoskeleton is, in the words of the textbook, an intricate network of protein filaments that extends throughout the cytoplasm. Fair enough, that's what the cytoskeleton is. And what we see here is a fluorescent microscopy image of a cell where the three components of the cytoskeleton have been labeled with three different fluorophores, with three different colors. Well, and I should say four fluorophores because that nucleus is stained with a dye called DAPI, which stains the chromosomes, the DNA. So the nucleus is in blue. In green are the intermediate filaments, and you can see that they diffuse and spread all throughout the cell, giving the cell structural stability. In yellow, and if you look closely enough, you can see some thick yellow bars scattered here and there, are your microtubules. And then in red is actin. And again, actin we'll talk about on Wednesday, or the video for Wednesday. Although some cytoskeletal proteins and cytoskeletal components can be found in bacteria, they aren't a true cytoskeleton. True cytoskeletons are solely eukaryotic. They're one of the defining features of a eukaryotic cell. And unlike a skeleton, sometimes that word is misleading there. The cytoskeleton, the cell's quote unquote skeleton, is highly dynamic. I mean, bones are dynamic too. We have osteoblasts. There are cells that break bone down and build new bones. So I don't want to make it sound like bones are completely static. But once you're an adult and you have your skeleton, the changes to the bones of the skeleton are fairly minor. Not so for the cytoskeleton. The cytoskeleton is constantly in motion. It's very dynamic. It's responding to changes in cell signals. It's responding to changes in cell state. It's responding to changes in cell need. And that's what we'll talk about all week. So what this really means is that the cytoskeleton is not just the bones of the cell, it's also the muscle of the cell. And indeed, whenever a cell is in motion, Whenever a cell is moving, like an amoeba cell that's blobbing somewhere, one of our own fibroblast cells that's crawling to a new location, muscle cells contracting, mitosis, where chromosomes are being pulled to opposite sides and the cell is actually constricting in the middle so that it can split into two. All of these actions are cytoskeletal. The cytoskeleton is responsible for all of that. Without a cytoskeleton, all cellular mobility would cease. There would be no more motion of or in a cell. In addition to this, the cell can be thought of in many ways as an ATP factory. Making ATP is a priority for the cell because we got to bribe the universe because of all this damn order. And to get away with all this anti-entropy, we have to bribe the universe with a whole lot of heat. So making ATP is a priority for the cell, and that's why we spend so much time on cellular respiration. Like any good, well-designed factory, even a cellular factory for making ATP, the interior of the factory needs to be very compartmentalized and organized so that things can occur in a very systematic way, and such is the case for the cell. It's the cytoskeleton that controls that compartmentalization and that organization. The cytoskeleton is responsible for everything in the cell being in its right place. The cytoskeleton is also responsible for intracellular transport. Now, we've talked a lot about intracellular transport, right? When we talked about vesicle formation, proteins that are processed by the Golgi bind to a cargo receptor. Cargo receptors are gathered by adaptins. Adaptins recruit clathrin. Clathrin makes the pit. The pit gets bigger and bigger. Dynamin severs the vesicle from that membrane. We've got RABs in the membrane of the vesicle. We've got V snares in the membrane of the vesicle. And I don't need to tell you the rest. The vesicle makes its way to the target membrane, and we've got docking and fusion and cargo delivered. Well, like I just kind of hit you with a black box of magic there, right? Whole lot of detail, and then the vesicle gets to where it's going and the cargo's delivered. How does that happen? How does the vesicle actually get carried to its destination? Well, that all happens through the cytoskeleton. In fact, we'll talk about that today. Here again, we've got a DAPI stain, so the chromosomes are stained in blue. The microtubules here are stained in green. This is a cell in the middle of mitosis, and so get ready for it. It's happening. Mitosis all over again. We're going to go deeply into mitosis next week from a cellular biology perspective. But those are microtubules in green. And what are those microtubules going to do? They're going to separate those sister chromatids. We're looking at a metaphasic cell. This is a cell in metaphase where the chromosomes are lined up in the center of the cell. And those microtubules are going to pull those sister chromatids apart, moving us into anaphase. So cytoskeletal components pull the sister chromatids apart, microtubules specifically. Then we'll go into telophase, and after telophase, we have cytokinesis. Cytokinesis involves the constriction of the center of the cell so that eventually that one cell will split into two. Well, that's also due to the cytoskeleton. In fact, it's due to actin of the cytoskeleton. So the proteins of the cytoskeleton are formally, I'm getting ahead of myself, formally three different proteins. And each of those proteins is specialized for a different role in the cytoskeleton. We have intermediate filaments. Intermediate filaments are purely structural. They're like protein rope. They help hold the cell together and they allow the cell to withstand tensile stress, meaning stretching or compression. We've got microtubules. Microtubules are the fattest, widest, with the greatest diameter of all of these cytoskeletal components. And this is what's key. If something is in motion within the cell, microtubules are involved. So microtubules are needed for things to move inside the cell, vesicles to get to where they're going, sister chromatids to be pulled apart, organelles to be correctly placed and organized. If something is moving in the cell, it's moving because of microtubules. And then the thinnest of the three, with the smallest diameter, are actin filaments. They can also be called microfilaments because they're so thin. Again, actin filaments will be the focus of our next video that we watch for Wednesday in the class. But actin filaments' dedicated role is cellular motion. If the cell itself is in motion, actin filaments are involved. And so to highlight the difference between microtubules and actin filaments, if things are moving in the cell, microtubules are needed. If the cell itself is in motion, actin filaments are needed. So perfect example, and one that we'll go into in some great detail in the next video, is muscle contraction. In a muscle contraction, a muscle cell is getting smaller, it's getting more narrow. The cell is moving, and that's due to actin. Not only is each of these cytoskeletal components different in its specialization and the job that it does in the cell, but it's also different in its construction and what it's actually made of. And each of these cytoskeletal components is made up of different protein subunits. In fact, for each of them, it's a family of similar proteins that come together to make intermediate filaments, microtubules, and actin filaments. The family of proteins that make microtubules are called tubulin, and there are two main types, alpha and beta. There's also a gamma that we'll discuss briefly. The microfilaments are made up of actin, that's why they're called actin filaments. And we're going to go much into depth of the intermediate filaments in just a second. The intermediate filaments are the most varied, the widest and broadest of protein families for cytoskeletal components. In every case here, for all of these cytoskeletal components, proteins come together by the thousands to create long fibers of protein complexes. So again, as we start thinking about tying threads of this course together, we are deep, elbows deep in quaternary structure, where different polypeptides are coming together and associating to make a much larger structure, a complex. These fibers, whether they're intermediate filaments, microtubules, or actin filaments, are then added to the overall meshwork of the cytoskeleton, and the cell can behave properly. So that's the setup for the cytoskeleton in general. And now we can start getting into intermediate filaments in detail. So as I mentioned before, the specialty of intermediate filaments is what we refer to as tensile strength, the ability to stretch, be pulled, or be compressed and squeezed without deformation, without any structural damage. So the main function of intermediate filaments seems to be to help the cell withstand essentially mechanical stress. Again, in diameter, in thickness, the intermediate filaments are between the microfilaments, which are the thinnest, and the microtubules, which are the thickest, and that's actually where they get their name. That's why we call them the intermediate filaments, because they're intermediate in diameter. Intermediate filaments are also the toughest and the most durable. In fact, they're the only of the three cytoskeletal components whose sole job is structural. Actin and microtubule are both extremely functional in what they do. In most cells, intermediate filaments are found in the cytoplasm, where they form a network of these rope fibers. They surround the nucleus and they extend outward towards the cell membrane. If you remember that image I showed in the earlier slides where the intermediate filaments were fluorescing green, you see them all throughout the cytoplasm. And you see that a little bit cleaner right here. Here we have one stain that is just staining the edge, the periphery of the cell membrane to delineate one cell from another. That's like a light blue, green. Would we call that aqua? Would we call that teal? Maybe I should make that an Edpuzzle question. But anyway, that delineates the cell. But what we see in green are the intermediate filaments. And again, you can see the intermediate filaments are really taking up a large volume of the cytosol, and they're giving the cell its structural integrity. Intermediate filaments are often anchored on either end. One end is typically anchored to the outer surface of the nuclear membrane, and you can kind of see that here in the image. And then the other end is anchored to other components of the cell, sometimes the inner leaf of the cell membrane. Sometimes it's where one cell joins another. So intermediate filaments are involved in cell-cell contact points, which we will talk about in our last week of instruction. Intermediate filaments are also found in the nucleus, where they are the main component of the nuclear cortex. Ooh, I like that term, the nuclear cortex. Where have we heard the term cortex before? Let's pause the video and think about that for a second. We talked about cortexes before when we were discussing the cell cortex. And the cell cortex was a protein meshwork that lied under the cell membrane and gave the cell membrane, in fact, gave the cell its shape and structural integrity. Try to make a big deal of saying that the cell membrane has the consistency, viscosity, and durability of olive oil. And so we really don't want olive oil being our main structural component, and we don't have to worry about that because the cell cortex is a very durable protein cage that the olive oil cell membrane sits over. That's the cell cortex. The nucleus also is made up of a membrane. Nuclear membrane is also olive oily, and we want a little bit more structural integrity for the nucleus as well. And so the nucleus has its own cortex, called the nuclear cortex, And the nuclear cortex is made up of intermediate filaments, in fact, proteins that are referred to as the nuclear lamina. So this diagram you're looking at here is not a diagram of the cell. This is a diagram of the nucleus, believe it or not. The outer surface of this diagram is the nuclear membrane. We see our nuclear pore complexes studying that membrane. Remember, these are the things that the nuclear transport receptors can move through by pulling on those fibrils and opening that cage. We talked about protein transport into the nucleus. And then we see the interior of the nucleus is extremely organized. In fact, it has its own internal structure called the nucleolus, where specific genes for specific functions are stored. But the nucleus itself is highly organized. It's almost like its own, well, I don't want to say like its own mini cell because that's a little bit too much, but it's a very organized structure in its own right. But here we see it underlying the nuclear membrane is this crisscross protein cage of the nuclear cortex. And this is what gives the nucleus its shape and structural integrity. This is what holds the membrane up into a roughly sphere-like shape. And that nuclear cortex is made up of the nuclear lamins, intermediate filaments, giving that structural stability. I also want to say before we move on to this that regardless of how well you do in your courses, how much you ace your college curriculum when you do graduate, whether that's in a couple weeks or a year in a couple weeks, regardless of how much you retain from the information that was shared with you over your 4 years or so of college, just take a moment to appreciate how little you know about the discipline you've studied, how little biology you actually know. You're never going to be taught any of the depth or complexity of the interior of the nucleus. It just doesn't belong in an undergraduate curriculum. And let me not kind of point fingers at you. I've got a lot more training in this discipline than you do. It took me about 6 years to get my PhD, and I've been teaching this stuff for a long time. I think it's fair to say I know more biology than you do, and I should because I'm being paid to teach you. But there's so little I know about this discipline. I mean, I would be thrilled to learn that I know 5% of what the discipline of biology has to offer. Now, to be fair, I know that 5% really, really well. There's so much of biology I just don't know. We know it, but I don't know it. So it's nice sometimes. It's humbling, but it's nice to know that there's so much more out there to learn if only we have the desire and the passion to learn it. So the nuclear cortex, made up of intermediate filaments, is there to strengthen the nuclear envelope, give that structural stability to the nuclear membrane. So some of the function of intermediate filaments, right? Some examples of what intermediate filaments do. What's the anatomy of intermediate filaments? How are intermediate filaments made? Well, what intermediate filaments are made of and the nature of their construction contribute to their job, contribute to the tensile strength that they offer. Intermediate filaments are made like rope. And that's not an analogy. That's not me trying to say like, oh, like kind of think of rope and nope, nope. Intermediate filaments are made like rope, period. Here's a close picture of rope. And let's take a good look at that picture. Rope is made up of very, very thin fibers that are twisted together. So we see some of those thin fibers kind of poking out here and poking out here. So those thin fibers are twisted together to make these fibers, right? These fibers are bundled up together to make this fiber, and then two or more of these big fibers are braided together to make the rope. So rope is a bundle of fibers that were bundled into bundles that were braided. That's rope. And that construction gives rope enormous tensile strength. If you think of what rope is supposed to do, rope is supposed to stretch without snapping. Rope is supposed to stretch without tearing. Whether you're lowering someone down into a well, you're rock climbing up and you have rope as a support, you're hauling something in the backyard using a pickup, whatever you're doing, if you're using rope, your intention is to connect two things together and have an enormous amount of force on those two things in opposite directions, and the rope is going to hold them together and stretch without snapping. And that's exactly what intermediate filaments are meant to do, hold things together and stretch without snapping. And so intermediate filaments are built just like rope. To make intermediate filaments, pairs of long linear intermediate filament subunit proteins first wrap around each other to form a dimer. So here's one long protein subunit of intermediate filament. Here's its amino terminal domain. Here's its carboxy terminal domain. So the protein is going from N to C, and it's mostly made up of alpha helices. That's one subunit. We take two subunits and we twist them together. Then we take this twisted dimer and we stagger it with another to make a tetramer, right? See how we're like mimicking rope here? We keep just bundling these fibers together. So it's called a tetramer because tetra is the prefix for four, and now we've got four subunits involved. Then we take these tetramers and we bundle them together to make a larger bundle of fibers. And then we take that larger bundle of fibers and we do exactly what we do with rope. We twist it together. I'm saying we, the cell twists it together. And so this is a complete intermediate filament right here. But let's look closely. An intermediate filament is nothing but a bundle of a whole lot of smaller filaments that themselves are bundles of tetramers, tetramers that are bundles of dimers, and dimers that are twisted bundles of single monomers. Small fibers twisted together into larger fibers, twisted together into larger fibers, twisted together into an intermediate filament. And do I need to show it again? Small fibers twisted together into larger fibers, twisted together into larger fibers that are twisted together into rope. Same job, same structure, because structure equals function. The intermediate filament protein heads, those are the globular regions on the amino terminal end and the carboxy terminal end, are used as anchoring points. They attach to many different things. Here, in the case of the tetramers, they attach to other tetramers to allow the fiber to get really long. So these globular domains can interact with other intermediate filaments to get the fiber to be long. But we also have globular heads on the very ends of the intermediate filament, one end and the other. And those globular heads can be used as anchoring points to adhere to the outer leaf of the nuclear membrane or adhere on the other end to some other structure in the cell, such as organelles. Intermediate filaments, just like rope, provide the cell with strength and resistance to mechanical stress. We find intermediate filaments to be most abundant in cells that experience the most mechanical stress. Cells that are designed to stretch, such as epithelial cells on the skin, muscle cells that contract and relax and stretch. And we also find intermediate filaments in the most sensitive to mechanical stress cells, like the axons of neurons. Right, if you think of the axon of neurons, which by the time you're watching this video, we have talked about 2 weeks ago in class. If you think of the axons and neurons, they're very, very long, they're very, very thin, and they're pretty much just membrane, super prone to breakage. But if you load that axon up with intermediate filaments, you give it tensile strength. You allow that axon to stretch without tearing. And that's what intermediate filaments do in all of these cell types. They stretch, they distribute the force of that mechanical stressing, that stretching, throughout the entire cell, rather than having those stresses being local and leading to cell tearing. So if you were to stretch a bunch of cells without intermediate filaments, they would break. I mean, what's holding them together? Olive oil. But if you load those cells with intermediate filaments that are anchored to the nucleus and anchored to the cell membrane, now the tissue, now the collection of connected cells can stretch and not snap. That's the tensile resistance. Without the distribution of forces that intermediate filaments give, cells tear. And again, this is not an analogy. We do this. The whole idea of carbon fibers in fiberglass, the whole idea of reinforced concrete, putting steel rods. Well, what are rods? Rods are fibers of steel bundled together that are bundled together. I mean, it's steel rope. That's what a steel rod is. It's steel rope. We put fibers into our building materials that need to withstand mechanical stresses. We put carbon fibers in fiberglass and steel rods in concrete so that these buildings and these structures can deform and stretch and stress without tearing, without giving way. Getting more into the nitty-gritty of intermediate filaments, there are four main classes of intermediate filaments. The first one you've probably heard of before, it's keratin. Keratin filaments are in epithelial cells. These are the intermediate filaments that represent the broadest category or type of intermediate filaments. They're found in the cytoplasm of most every single cell of any vertebrate, specifically in the lining cells of the epithelium. Specialized forms of keratin, and probably the context you've heard it in, make up things like hair, fingernails, feathers, claws of other animals. That's the keratin that you may have heard of. Each keratin intermediate filament is made up of a mixture of different specific subunits, but those subunits are assembled in just the way that I said before. Two monomers come together and twist to form a dimer, dimers make tetramers, tetramers make fibers, fibers bundle together make the intermediate filaments. But because we have different types of subunits that can be involved in keratin formation, keratins are also the most diverse class of intermediate filaments. Keratins do what all intermediate filaments do. They distribute mechanical stress of stretching, especially in the epithelial cells, in our skin, which stretches as we move, in our gut as we eat and the stomach stretches or the intestines stretch, in our lungs as we breathe in and breathe out, and our lungs expand and contract with the filling and losing of air. Epithelial cells need to stretch in the context of their jobs, in the context of their work, and intermediate filaments, specifically keratins, allow that to happen. We can see a case study of this in the unfortunate case of epidermolysis bullosa simplex. That's a genetic disorder, a rare genetic disorder that involves mutations in the keratin gene. So the genes that encode the keratin protein are mutated. Keratin proteins are not bundled properly, and so the keratin intermediate filament family does not assemble properly. And as a direct result of this one mutation, skin is very vulnerable to mechanical injury. Skin tears very easily when stretched. There's a lot of bruising and tissue damage, even with gentle pressure. You might remember in class a couple weeks ago, I had us kind of push on our skin as a view of sensation. If you had epidermolysis bullosa simplex, you'd have a black and blue from that gentle pushing, just from damaging the skin with that very, very light mechanical stress. Okay, now just give me like 30 seconds to go on my soapbox here and rage a little bit. This is the stupidest stuff I've ever seen in my life. All these beauty products that have keratin in them and tell you that the keratin is going to make your hairs healthier, or you can put keratin polish on your nails and your nails will get so ridiculous. The keratin needs to come from the cells. The keratin needs to be secreted by the cells in a complete form, built as a bundle. Simply exposing your hair to keratin doesn't help the hair get stronger. The keratin that you are exposing can't incorporate itself into the keratin of the hair. That keratin needed to be made in the cell and secreted as an extracellular protein. In addition, sometimes I've seen keratin or collagen supplements where you swallow a pill that contains these supplements and is supposed to make your skin healthier or younger or your hair healthier. The first thing that happens to that keratin or the collagen when you swallow it in pill form is it's going to be digested into individual amino acids. And will some fraction of those amino acids be used to make the keratin and collagen that your cells need? Yeah. But the same is true of everything else you eat. It's going to be broken down into individual amino acids. So if you're falling into that trap of buying these fancy shampoos that contain keratin, don't. They don't. Exogenous keratin has no effect whatsoever on anything. Now, the shampoo might be really good and make your hair smooth and feel nice, like we see in this picture. It might be a high class shampoo because of everything else that's in it. But trust me, it's not the keratin that's making your hair any healthier. Hopefully, that was about 30 seconds. So first class of intermediate filaments, keratin. Keratin providing tensile strength to epithelial cells. Second class, vimentin. Vimentin and vimentin-related filaments is the second class of intermediate filaments, and we find vimentin in connective tissue, muscle cells, the support cells or glial cells of the nervous system. So not in the epithelial cells, not on the surface cells of our body, but on the connective tissues of our body. Third class are neurofilaments, kind of self-explanatory. This is a dedicated class specifically for neurons. They provide the structural support of axons, so axons do not tear because axons are the most fragile part of a neuron. They also seem to play some role in neuroregeneration. Species of animals that can regenerate severed nerves, especially species of animals where like you can sever a limb and the limb will regenerate. They have very different neurofilaments, which has led a lot of researchers to wonder if the neurofilaments that we have is one of the limiting factors as to why our peripheral nervous system can't regenerate that efficiently and why our central nervous system can't regenerate at all. In fact, one of the labs I did a rotation in the first year of my graduate training was studying just that. It was a really interesting project. So there's a lot of potential there to perhaps have new therapies for uh, injuries that lead to quadriplegic and loss of sensation. If we could figure out neurofilament stem cell therapies, maybe we can coax these neuronal cells to regenerate. And then the fourth class we talked about already, that's the nuclear lamins, the proteins that are involved in making the nuclear cortex. These proteins strengthen the nuclear envelope and give structure to the nuclear membrane of animal cells. So the four classes, keratin, vimentins, and neurofilaments are all cytoplasmic, giving strength and stability to epithelial cells, connective cells, or neuronal cells, respectively. And then we have the nuclear lamins as the fourth class. They're the only class that's not cytoplasmic, and they're making up the proteins of the nuclear cortex. Okay. So again, the first three classes that I just talked about are cytoplasmic. Only the lamins are nuclear. And the first three classes of intermediate filaments on the previous slide are also made like rope in the way that I discussed at the very beginning. The nuclear lamins are constructed in a rope-like way, but they don't end up being long fibers of rope. Instead, they wind up being a net of rope. You can think of like a cargo net that you might climb at a amusement, not amusement, but at a playground, or a cargo net that you might see for shipping or fishing. So still rope, but rope that's bundled together into a two-dimensional mesh. So you're actually looking here at an electron micrograph, a picture taken with an electron microscope of the nuclear cortex. So these are the nuclear lamins bundled up in just the way I saw. And you can see how much like a cargo net it looks like, right? It's pretty interesting. And so this makes the nuclear lamins unique, that they don't make long linear rope, but they make cargo nets of rope. Additionally, the nuclear lamins, unlike the other three classes, are extremely dynamic, meaning they disassemble and reassemble very often. And in fact, they disassemble and reassemble with each round of mitosis, with each round of cell division. And they have to do that. So again, I don't want to like trigger anybody. I know a lot of people just don't like mitosis. I'm not sure why, but I know a lot of people don't. I'm going to bring you back to Bio 107 or bring you back to any other more recent class that you might have talked about mitosis. I'm also planting seeds for next week when we're going to talk about mitosis quite a bit. Just as a reminder, I proudly prefer milk and tea, interphase, prophase, prometaphase, metaphase, anaphase, telophase, right? Those are the stages of mitosis. And the hallmark defining feature of prometaphase is the breakdown of the nuclear envelope. The nuclear membrane breaks down in prometaphase so that the microtubules can attach to the sister chromatids in metaphase and line them up in the middle. So, like, what did I just say? I said in prometaphase, the nuclear envelope has to break down. Well, for the nuclear membrane to break down, that means the nuclear lamins have to break down because they are the protein cage of the nuclear membrane. So unlike all of the other classes of the intermediate filaments, the keratins, the vimentins, the neurofilaments, nuclear lamins must disassemble and reassemble with each round of mitosis. And this disassembly and reassembly is controlled by phosphorylation, which means it's controlled by kinases and phosphatases. So we might have an intact nucleus with its intact lamins giving a nice strong nuclear cortex. And then there is some signal that's received that it's time to enter prometaphase and the nuclear membrane needs to break down. A kinase is going to become active in response to that signal. That kinase is going to phosphorylate the nuclear lamins, and due to that phosphorylation, the nuclear lamins are going to become weak and they're going to disassociate from one another. The nuclear cortex is going to break down, and once the cortex isn't there, there's no structural integrity of the nuclear membrane. The nuclear membrane dissolves. Well, that's good because microtubules will then attach to the chromosomes. We'll line the chromosomes up in the center. We'll pull the sister chromatids apart. We get each set of chromosomes to opposite sides of the cell. And now it's time to make new nuclear membranes around each of those sets of chromosomes. That's the hallmark feature of telophase, creating new nuclei around each bundle of chromosomes on either side of the cell. Well, how are we going to facilitate that? We're going to activate a phosphatase to dephosphorylate all of these nuclear lamins, and then they will spontaneously reassemble into nuclear cortices and will make two new nuclei. So the breakdown and reassembly of the nuclear cortex is governed by the action of kinases and phosphatases, respectively. What's turning the kinases on at the right time, you ask? What's turning the phosphatases on at the right time, you ask? Why are these kinases and phosphatases active only in prometaphase and telophase, respectively? You just asked? Well, I'm glad you asked, because we're going to be talking all about that next week. So, to wrap up intermediate filaments for now, many of the intermediate filaments are stabilized and reinforced by accessory proteins. So they can't do their job all on their own. And one such example of an accessory protein is plectin. Plectin is one example of many, many accessory proteins. And what plectin does is cross-links intermediate filament ropes together into bundles, into more cargo net-like three-dimensional arrays, into stronger arrays of tensile strength. These accessory proteins, like plectin, also link intermediate filaments to other components of the cytoskeleton, like microtubules or actin, allowing the cytoskeleton to behave as one single structure, providing even more strength against mechanical stress. The importance of these accessory proteins is also illustrated by mutations. One mutation that I won't name here because we don't need the name, we've got enough terminology so far, but one mutation to the gene that encodes just the plectin protein causes a devastating skin disorder that's even worse than epidermolysis bullosa simplex. This mutation in plectin causes very similar symptoms to epidermolysis bullosa simplex. We've got easy breakage of the skin, easily bruising with mechanical stress, but it also has symptoms of muscular dystrophy because neurofilaments are involved and neurodegeneration. In fact, mice without the gene for the plectin protein die. So that emphasizes just how important these accessory proteins are. Okay, so let's move into the second half of the video and talk about microtubules. Microtubules play a crucial role in organizing this eukaryotic cell. And again, just as a reminder and as an overview, if things are moving inside a cell, microtubules are involved. Microtubules are long, stiff, and hollow, and they're tubes of protein, of course, and they're very dynamic. Unlike intermediate filaments that are fairly static once they're made, except for the nuclear lamins, remember the nuclear lamins are the exception, but unlike the other intermediate filaments that are fairly static and stable when they're made, microtubules are highly dynamic. They rapidly disassemble at one location, they reassemble at another locations. Also, unlike intermediate filaments that just kind of self-assemble, microtubules require a point of origin. They have to be made at a starting point. And the starting point for all microtubules is the centrosome. You may have heard of the centrosome before. You certainly did if you had me from Bio 107. The centrosome plays a key role in mitosis because all microtubules originate from the centrosome. In fact, in this stylized image from a movie that we're going to watch near the end of the semester, a very short movie, we see the centrosome here, and all of these projections coming out of the centrosome are microtubules. This is a centrosome, and this is a centrosome, and all these green fibers are microtubules. So all microtubules originate from the centrosome. So this is our second kind of mitosis trigger warning. We'll be talking much more about this next week, but this is where the microtubules, the spindle fibers that pull cystochromatids apart, come from. So once made, microtubules extend out from the centrosome, and they head generally outwards. Usually the centrosomes are near the nucleus, and the microtubules head outwards towards what we refer to as the cell periphery, in the direction of the cell membrane. And then they anchor, and they create protein tracks throughout the cell, tracks on which transport will occur. Very briefly mentioned when we were talking about vesicle transport, these walking proteins, proteins that would walk along and carry vesicles from the origin to the target. Well, these walking proteins are physically walking on tracks of microtubules. That's how vesicles get to where they're going. The vesicle is made in the way we already described, and the vesicle is going to be carried and transported by specific proteins that we'll introduce in just a few slides but those proteins are walking on microtubule highways. Microtubules also anchor and localize other organelles. Again, anytime anything's moving in a cell, even if it's moving to just be organized, to put the mitochondria where they belong, to put the ER where it belongs, to put the Golgi where it belongs, that's movement, that's organization of organelles, that's going to be mediated by microtubules. And so unlike intermediate filaments, which are very, very structural and don't really have a function other than tensile strength, microtubules and actin filaments are much more functional than they are structural. They do things in the cell, and they allow the cell to do things itself. For mitosis, the cytoplasmic microtubules that are serving as the highways and roadways for vesicle transport, all of those well, most of those microtubules need to be broken down and disassembled. Because if we're in mitosis, we need to rebuild our microtubules into the mitotic spindle, into the cables that are going to grab onto sister chromatids and pull those sister chromatids apart. Then once mitosis is done, we got to break down those mitotic spindles and rebuild them into the highways and roadways of the cell so that vesicle transport can occur. And then as we enter prophase again and another round of cellular division, we got to break down those roadways and highways and rebuild them as mitotic spindles. So microtubules, at least a subset of microtubules, are very dynamic, being broken down and rebuilt, depending on where in the cell cycle, what stage of mitosis the cell is in, if it's dividing. Now, that's not true for all microtubule-based structures. Some microtubule-based structures are quite static. Examples of this would be flagella and cilia. These structures that are needed for cellular motion, like swimming organisms or gametes, male gametes in most animals, it's microtubules that are allowing that motion to occur. Microtubules make up flagella and cilia, and those microtubules don't get broken down for mitosis. But the microtubules that are all throughout the cell, connecting different parts of the cell, are very, very dynamic. So that's the function of microtubules, following the same structure we did for the intermediate filaments. Now that we talked about structure, let's talk about anatomy. Let's talk about how they're made, what they do. Microtubules are made up of a subunit protein called tubulin. So microtubules are made up of tubulin. And tubulin proteins themselves are dimers of two similar but different globular proteins. And we call those similar but different globular proteins alpha tubulin and beta tubulin. So how do we make a long fiber protein out of globular spherical subunits? Well, we do it by stacking these dimers together, one next to the other, next to the other, next to the other, to make a protofilament, a chain of tubulin dimers that makes a long protofilament. And then we put 13 protofilaments around each other, side by side, and doing so, we create a tube or a hose. So this dark green and this light green make up a tubulin dimer of alpha and beta subunits. And we just stack those dimers together to make a protofilament. And then we get 13 protofilaments and we put them together as a round ring, and we've got ourselves a microtubule. This is a microtubule as if though we're looking at it head on, and then this is the microtubule in cross section. And I'm going to start counting at the one that has the line through it here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilaments side by side create this fat, thick, hollow microtubule. Again, the two different categories or types of subunits for tubulin are alpha and beta. Please don't get this confused with secondary structure. I'm not saying that one subunit is made up of helices and the other subunit is made up of sheets. These are globular proteins. They all contain secondary structure and tertiary structure. They're made up of helices and sheets. We've just named one type of tubulin protein alpha and one type of tubulin protein beta. As I showed you in the schematic, the way that these protofilaments are formed is that they are stacks or chains of alpha beta subunits. So that means the pattern of subunits in any protofilament is alpha beta, alpha beta, alpha beta, alpha beta, which means this ring on one end of the microtubule, where's my cursor? This ring on one end of the microtubule is going to be nothing but alpha subunits, And on the all the way other side of this microtubule, on its other end, the ring is going to end with beta subunits. This means that microtubules have polarity. They have two different ends. The beginning end of a microtubule is its alpha end, and the end of the microtubule is its beta end. This is true of the protofilaments individually, and so it's true of the entire microtubule. Again, microtubules have polarity. They have two different structural ends. And to keep things simple, for microtubules, we call one end the plus end and the other end the minus end. The alpha end, the beginning of the microtubule, is its plus end, and the beta end, the end of the microtubule, is its minus end. And this diagram just shows you true electron microscopy pictures of microtubules, both looking at it from the side, and you can actually see, not only can you see the protofilaments, you can see a hint of the spherical structure of the individual subunits. That's pretty amazing. And then here is that microtubule in cross section, just as we see here, with 13 tubulin monomers in a ring. It's beautiful, gorgeous stuff. As microtubules form in a living cell, these tubulin dimers can be added to one end of the growing fiber or the other. So we can conceivably and theoretically add tubulin dimers to the plus and minus end. However, the dimers are added much more efficiently to the plus end for reasons that we'll see in just a second. This difference in end and this difference in growth is crucial to the function and structure of microtubules because of what microtubules do, what they serve as. They serve as the highways of the cell. And proteins are going to walk along these microtubules to deliver cargo. And for proteins to walk along these microtubules and deliver cargo, those proteins need to know in which direction they're moving. Just like roads have directions, the right lane is heading in one direction, maybe south, which means the left lane is heading in the other direction, maybe north. And you need to know what end of the road you're on so you know what direction you're going in as you drive along it. Same is true for microtubules. Having two different ends allows these walking proteins to walk their cargo in the right direction. The two proteins that do walk along microtubules are called dynein and kinesin. We'll talk about them in just a second, but they walk in opposite directions. And in order to successfully and functionally walk in opposite directions, dynein needs to know that it's moving towards the negative end because that's the direction it needs to walk in. And kinesin needs to know that it's walking towards the positive end. So the ends must be different. Textbook says this perfectly. The textbook says if microtubules had no polarity, they couldn't serve their function in defining the direction for intracellular transport. Cargo would not know where it was headed if microtubules did not have polarity. So how does that polarity arise? How do microtubules actually get made? And what the hell is a centrosome? All of those answers come together in the story of the assembly of microtubules. Microtubules form rapidly and efficiently when tubulin dimers are being added to an already existing microtubule fiber. Very difficult for microtubules to start being made from scratch. It's much easier for microtubule dimers, tubulin dimers, to be added to an already existing tube. And that's what centrosomes do. Centrosomes serve as that starting tube. They serve as tubulin organizers, coaxing alpha beta tubulin dimers to form protofilaments and eventually form a microtubule itself. Once those protofilaments begin being formed and once tubulin dimers are added, more tubulin dimers will join more easily and more tubulin dimers will join more easily and a short tube will become a long tube and the process will just kind of snowball. It's getting it started that's difficult, and that's where centrosomes come in, starting this assembly. Centrosomes, if you can get a really close look at them, are studded on their surface with thousands of ring-shaped structures. And those rings on the surface of centrosomes are made up of a tubulin subunit. But it's not alpha or beta tubulin, it happens to be gamma tubulin, and gamma tubulin is only found on the surface of a centrosome. But alpha beta dimers love interacting with gamma tubulin. And so you will get dimers begin to form protofilaments on these gamma tubulin rings right on the surface of a centrosome. This is how centrosomes serve as the nucleation site of new microtubule formation, how they serve as the origin point for microtubule formation. Again, once you start making a short tube, that short tube is going to get longer all on its own, and we see that we'll just keep making these microtubules out from the centrosome. Centrosome is the origin point. The negative end of the microtubule is embedded in the centrosome. It's beta tubulin. Let me get that right. Hold on. Yeah, it's beta tubulin that interacts with gamma. And so the negative end of the microtubule is embedded in the centrosome, and the positive end of the microtubule reaches out towards the cellular periphery in the direction of the cell membrane and the organelles that are further out in the cell, in the cytosol. One interesting thing about microtubules that contrasts it dramatically from intermediate filaments is this idea of dynamic instability. Yes, once a microtubule has been started at a centrosome, it's going to get easier and easier to add new tubulin dimers to the growing plus end. That's true. And that process is going to go on for minutes. But then, without warning and spontaneously, many microtubules will spontaneously disintegrate. They'll collapse, they'll break down. Those tubulin dimers will begin falling off the positive end, and that will cause the microtubule to shrink backwards and fray. Sometimes these microtubules only partially fall apart. Sometimes they fall apart all the way back to the gamma tubulin ring at the centrosome. But this flux of build, disintegrate, 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 make a long microtubule, and then the microtubule spontaneously breaks down again. That's what we mean by dynamic instability. Dynamic meaning in motion or always changing, and instability meaning it's not stable. The microtubule is not going to stay put. Think of how different this is from intermediate filaments. Intermediate filaments are protein rope. You make an intermediate filament, you braid all these things together, you wrap all these fibers together, and you've got yourself a very thick, stable rope that's not going anywhere. Microtubules are the exact opposite. Microtubules fall apart all on their own. And that's just nuts. But there's a reason for it. Cells don't do things like this unless there's a good reason for it, and the reason couldn't be any gooder. Each alpha beta dimer is capable of binding GTP or GDP. Don't tell anybody else I said this, but you can kind of sort of think of tubulin dimers as G proteins. When these tubulin dimers are bound to GTP, they readily bind protofilaments and add to the growing on the plus end. GTP-bound tubulin dimers efficiently join the growing ends of microtubules. So when we see a microtubule growing, it's growing because tubulin dimers containing GTP are adding themselves onto the growing end. And in fact, we call this a GTP cap. And those microtubules, I'm sorry, those tubulin dimers are very stable. They want to be there because they're bound to GTP. But like G proteins, the GTP that tubulin is holding is hydrolyzed on a timer. And after some time, the GTP will be hydrolyzed to GDP. And tubulin dimers that are bound to GDP don't really like being part of a microtubule, and they'd rather not be there. So as we look at this diagram a little bit closer, we'll see that all of these tubulin dimers with the red circles, they're bound to GTP. They're happy being on the end of the microtubule as it builds and as it forms. And this GTP cap is keeping all of these tubulin dimers in place. These tubulin dimers have already cleaved GTP to GDP. They don't want to be here anymore. But they have no choice to stick around because the cap is keeping the integrity of the microtubule in place. When microtubules are being rapidly polymerized, and we keep adding dimers containing GTP onto that cap, then as those later, or what I should say previous, as those previously added dimers begin hydrolyzing their GTP, and now they're holding on to GDP, they can't go anywhere because this cap is being extended too quickly. New GTP containing tubulin dimers are coming in, the cap's getting bigger. At the same time, these tubulin dimers are cleaving their GTP. They don't want to be there anymore, but tough luck because the cap continues to grow. So when microtubules are being rapidly made and the GTP cap is being consistently built and maintained, that microtubule is not going to collapse. And that's a good thing. The microtubule is growing. If the rate of addition of new tubulin dimers begins to slow down for whatever reason, and let's say it's taking some time to add new GTP-containing tubulin dimers to this end, it's taking so much time that these dimers have cleaved to GDP, these dimers have cleaved to GDP, we've only added a few new tubulin dimers, these dimers have cleaved to GTP, the GTP cap gets really, really small. Well, once the GTP cap gets too small because we've slowed down the rate of new tubulin dimer addition, we're going to have that microtubule break down. The GTP cap will no longer be able to keep those displeased GDP containing tubulin dimers in place, and the microtubule is going to disintegrate. It's going to start to fray. So when the rate of GTP hydrolysis catches up to the end of the GTP cap, because the new addition of tubulin dimers has been too slow, we're going to get the spontaneous destruction, disintegration, breakdown of the microtubule that will tip the balance towards disassembly. Yeah, and it's all because those tubulin dimers are bound to GDP. What's behind these tubulin dimers that contain GDP? Well, other tubulin dimers that contain GDP. What's behind them? Other tubulin. So once this process starts and these unhappy GDP-containing dimers begin to leave, we're going to get fraying often all the way back to the centrosome. The disassembled free tubulin dimers are going to hang around in the free tubulin pool. Eventually, they'll let go of that GDP and they'll pick up GTP. And once they pick up GTP, they're going to be very willing and ready to join a GTP cap and help some other microtubule grow. So the process can recycle and continue. Again, this is very different from intermediate filaments, where for intermediate filaments, stability was the main feature. For microtubules, dynamic instability is the main feature. Intermediate filaments are fibrous proteins that are built to be rope. Microtubules are globular proteins that are built to be highways. Intermediate filaments are remarkably stable, and microtubules couldn't be less stable. They're dynamically unstable. And intermediate filaments are purely for structural integrity and tensile strength. Microtubules have a job. They are the roads, they are the highways, they are the railroad tracks for transport, and they have polarity for that reason. So the striking differences in the structure and assembly of these two different kinds of cytoskeletal components highlights their different roles. No surprise, structure equals function, right? So of course that's going to be the case. So what we're really saying here is that microtubules are maintained by a balance of assembly, if that GTP cap is big, and disassembly, if the rate of new tubulin dimer addition starts to slow down. But ultimately, at some point, a microtubule has to stop being built, right? If a microtubule gets to where it's going, if it reaches its destination, we're not going to be adding any new tubulin dimers onto the cap. Eventually, all the tubulin dimers will have hydrolyzed their GDP. So how do microtubules ever stay stable? How do we stop microtubules that are going to the right place from falling apart? The short answer is that the plus end, the growing end, when it hits a destination that needs a microtubule bound to it, it will anchor. And once a microtubule is anchored on both ends, remember the minus end is anchored at the centrosome and the plus end is anchored at some target in the cell, the inner leaf of the cell membrane, the Golgi, the lysosomes. Once it's anchored to its target, that microtubule will be stable. And the only way it can be broken down and disassembled is for the anchoring proteins at the target membrane to pop and release that anchor point. So why? Why have this process? It seems remarkably inefficient, especially because it all hinges on a triphosphate, which we already know are very difficult and highly energetic to make. Well, the reason for this dynamic instability is because the cell has no way to send a microtubule out to a specific location. All of this microtubule construction is being done blind. So the process, the strategy is then the centrosome sends out microtubules in all directions all the time. And some subset of those microtubules, by random chance, like throwing darts at a dartboard blindfolded, some subset of those microtubules are actually going to hit an appropriate target. And when they do, The plus end will anchor at that target. Anchoring proteins will stabilize the plus end, and that microtubule will not break down. But for all of those other microtubules that didn't hit a target, all of the darts that didn't hit the dartboard because the cell is blindfolded, well, we want those microtubules to break down because they didn't find a target that they should bind to. And so we should retract those microtubules and try again. So the strategy makes sense. Again, here's the nucleus in brown, here's the centrosome. We're throwing out microtubules in every direction, like a blindfolded fisherman or fisherwoman, just putting lines out in every direction of the lake. Most of those microtubules are going to miss because they're being sent out blindly, just as much as those fishing lines aren't going to catch a fish. And those missed microtubules do and should retract back to the centrosome because they didn't attach to anything. But every once in a while, we'll get lucky and a microtubule will hit an actual appropriate target. And waiting for the microtubule at that target are anchoring proteins, here seen in purple, pink. And those anchoring proteins will latch on to the plus end of the microtubule and keep that plus end stable, even though those tubulin dimers are now bound to GDP. Those tubulin dimers don't want to be there anymore. But the anchoring protein does the job of the GTP cap and keeps those unhappy tubulin proteins from dissociating. So is this process random? Yeah. Is it energetically inefficient? Yeah. But the cell has no other way to do this because the cell cannot act with intention. This strategy allows the cell to set up this highly organized transport system without having the ability to intentionally build it. I'll say one other thing. This will come up next week, but keep in mind that this is great that we have all of these roadways and highways and railroad tracks for protein transport. But once the cell enters mitosis, we have to break all of those microtubules down into their individual tubulin dimers and then reassemble the microtubules into spindle fibers for cystochromatid separation. So if we think about that too, the most efficient way to have that occur very quickly is to have inherently unstable microtubules. When a cell leaves interphase and begins to enter prophase, and it's time to break down microtubules as roadways and build microtubules as spindle fibers, all that has to happen is those anchoring proteins pop at all the plus ends of those microtubules, and then those microtubules will disassemble all on their own because they were unstable to begin with. We can see evidence of this. Drugs that block microtubule destabilization. I know that's a double negative, but in other words, drugs that stabilize microtubules block mitosis. Mitosis can't occur if microtubules are stable, because in order for mitosis to occur, microtubules have to break down and be rebuilt dynamically for cystochromatid separation. So drugs that keep the anchoring proteins from popping, drugs that keep microtubules in place during interphase, block mitosis and cellular proliferation. So these make pretty potent anti-cancer drugs. Other drugs target microtubules as well. Some of those drugs block polymerization, but Taxol you may have heard of. Taxol has been a very well-received and high-efficacy cancer drug, and Taxol actually stabilizes microtubules, and doing so blocks cells from entering prophase. And of course, if a cancer cell can't enter prophase, it can't go to cytokinesis, it can't divide, and the only reason cancer is dangerous is because it's a disease of dividing cells. So before we begin to wrap up here, let's just talk a little bit about these motor proteins, these walking proteins that drive intracellular transport, and connect some of the material from, I think, 2 weeks ago to what we're talking about here. Like microtubules themselves, many cells have polarity. They have two distinct ends. And the most extreme example of that is neurons. We have a soma with dendrites on one end of the neuron, and then very far away down the axon, we have axon terminals on the other end. And those two ends of the neuron couldn't be any more different, structurally or functionally. Dendrites in the soma look very different than the axon terminal, and we receive messages at the soma, and we send messages through the axon terminal. So this polarity of the cell for a neuron is reflected in the polarity of the microtubule. And because the microtubule has two different ends, because microtubules are polar, this helps guide the transport of vesicles, in this case containing neurotransmitters, in the right direction. Neurotransmitters are made in the soma, and then they need to be shipped to the axon terminal. And the proteins that walk those vesicles from the soma to the axon terminal need to know they're walking in the right direction. And it's the polarity of the microtubules that allow them to do that. Again, we've talked about walking proteins here and there before, but finally we'll give them their due time. Let's stop calling these walking proteins. For first of all, walking proteins fall into a larger protein superfamily called motor proteins. Motor proteins move things, and they move things with conformational changes. In fact, the proteins that allow our muscle cells to contract are also motor proteins, but they use actin. And so we'll talk about them in the next video. Motor proteins use the energy from triphosphates, usually ATP, sometimes GTP, but they use the energy released from triphosphate hydrolysis to walk along, to travel along microtubules in today's case, or actin in the next video, and to do so in a single direction because they're following the polarity of the fiber. So let's orient ourselves to this image. This is a screenshot from a video that I'll show you near the end of the semester, but this large blue mass here is a vesicle. That vesicle is studded with Rab proteins and tethering protein. I'm sorry, Rab proteins and V snares, so that we can dock and fuse efficiently when we reach our target. This fiber right here is a microtubule, and this is our motor protein. The motor protein is bound to the vesicle on one end, and the other end of the motor protein walks. I kid you not, it walks. It walks along the microtubules in a specific direction. And that is vesicle transport. That's how vesicles, once they're formed, make their way to the destination for docking and fusion. There are dozens of different motor proteins that have been characterized so far, and there's probably more too that we haven't yet discovered, but we're going to focus on just a select few. These different motor proteins, they differ in the cytoskeletal component that they bind to, such as actin or microtubules. They differ in the direction they move. Do they move from the plus end to the minus end or the minus end to the plus end? They also can differ in the kind of cargo that they carry. The two examples that we'll focus on for microtubules and vesicle transport are kinesin and dynein. Before I go any further, I want to highlight here a potential point of confusion. We have talked about dynamin. Dynamin was the protein that severed a bud from a membrane. Dynamin causes vesicles to break off. That's not this. This is dynein. So dynamin separates vesicles, and dynein is a motor protein. Kinesins move away from the centrosome. They move from negative to positive. Kinesins carry cargo toward the cell periphery. Dyneins move in the opposite direction. They move towards the centrosome, which is towards the nucleus. Dyneins bring cargo deeper into the cell. So a quick little trick here. If you think about dynein and kinesin alphabetically, D comes before K. If you think about minus and plus, or negative and positive, minus negative comes before plus positive alphabetically. And so those two match. Dynein negative, kinesin positive. Both are in alphabetical order. Both of these proteins, kinesin and dynein, are dimers. They bind adenosine triphosphate, and they cleave that adenosine triphosphate to liberate the energy needed for this walking and to allow the walking to occur. The head domain, here's where it's just a little counterintuitive. What we refer to as the head domain of the protein is actually what's doing the walking on the microtubule, and the other end is what's bound to the vesicle, to the cargo. We call those the tails, not the feet, but the tails bind to the cargo. And this is a cycle. This cycle of triphosphate cleavage for motor protein function is going to weave itself all through this week. We're going to talk about a similar cycle in the next video for muscle contraction. But the cycle is bind ATP, cleave the ATP, making ADP, and then release that ADP. Those three things are going to lead to three different steps of walking. Bind ATP, cleave ATP, release ADP is going to result in the conformational changes needed by dynein or kinesin in order to allow this protein to walk along microtubules. So to put that in the context of the walking, this is kind of like the return state, and it's going to be step 1, step 2, step 3. Step 1 is lift a foot off the ground and move it forward. Step 2 is land that foot on the ground and adhere to the microtubule. And step 3 is drag the back foot forward. So lift your foot and move it ahead, plant that foot down and bind to the microtubule, and then bring the back foot forward. And then that brings you back to the starting point. You're ready to do it again. So if we think of that in terms of the triphosphate, when you bind ATP, it's lifting the foot of dynein or kinesin and just pushing that foot forward. That foot's still hovering in the air, you just extended the leg. Cleave that ATP to ADP that plants that forward foot down and sticks to the microtubule. Release that ADP, and now you're going to drag the back foot forward. You just took a step. Bind to a new ATP. Take this leg up. Where's my cursor? Lift this leg up and move it forward. Cleave that ATP to ADP, plant that foot down a little bit ahead, release that ADP, drag the back foot forward. Each of this is a cycle of steps. This is how kinesin and dynein walk. And if you want to, one, kind of see this explained in action, and two, see me make a bit of a fool of myself, probably, you can hunt down a video of me actually acting this out in my basement on YouTube on one of my channels. That was made during the pandemic, so I was teaching exclusively online. I couldn't do anything in the classroom, but I'll have a big gray, bulky Bruins sweater on. Bruins, don't get me started this season. But yeah, you can see me kind of pantomime this out. So when we say that cargo binds to cargo receptors are gathered by adaptin. Adaptin recruits clathrin. Clathrin makes a clathrin-coated pit. The pit gets bigger and bigger and bigger. We make a bud. Dynamin comes and severs the bud neck. We've released the vesicle. The vesicle is moved to the target where Rabs and tethering proteins and V snares and T snares can facilitate docking and fusion. Now that middle step isn't a black box anymore. How did those vesicles move from their origin membrane to their target membrane? They were bound onto dynein or kinesin. On the other end of that motor protein, it was bound to microtubules, and dynein or kinesin walked along those microtubules one step at a time using this ATP cleavage cycle to drag that vesicle to its target. And once the motor protein reached the target, Rabs and tethering proteins will dock, V snares and T snares will twist, and the cargo will be delivered. Now, this system of walking in microtubules isn't just for vesicle transport. It's also, again, for the organization of the organelles. So both the endoplasmic reticulum and the Golgi are positioned in the right spot and organized in the cell by interacting with motor proteins and microtubules. Kinesins attach to the cytosolic face of the ER membrane. Dyneins attach to the Golgi membrane face that's cytosolic. And then these walking proteins walk along the microtubules and stretch these organelle membranes out. So the ER and the Golgi are stretched out because they're dragged by motor proteins walking along microtubules. The best analogy I can give for this is what happens at a baseball stadium or on a baseball field when it starts to rain. So if you've never seen it before, they've got these giant tarps to cover the infield, to keep the infield from getting muddy. And a whole team of staff, they come out, each staff member grabs this tarp in a specific place, and then they drag this tarp over the entire infield. Well, in this metaphor, the staff workers are dynein and kinesin. The ground they're walking on is the microtubules, and the ER or Golgi membrane is the tarp. And the motor proteins are stretching that membrane out in the interior of the cell by walking along the surface of microtubules. We can see the importance of this because when we treat cells with chemicals that cause the disassembly of microtubules, and now dynein and kinesin have nothing to walk on to stretch those membranes out, the locations of these organelles change dramatically. The ER literally bounces back and collapses into a tiny little mass of membrane near the nucleus. And the Golgi fragments into a lot of small vesicles and completely loses its shape and integrity. So these motor proteins and microtubules are critical for cells to have the internal organization that they do. Interestingly, once these chemicals are washed out and removed, everything goes back to normal. The microtubules reassemble, the motor proteins have surfaces to walk on, and the organelles are reorganized. Again, cells are just amazing. So the cytoskeleton is far more than just the skeleton of the cell. The cytoskeleton is the structural and tensile strength of the cell and tissues. The cytoskeleton is the means by which cargo is moved around the cell and the facilitator of cellular organization. And the cytoskeleton is also the way that all voluntary cellular motion is possible. But for that piece of the story, you'll have to wait to the next video. What did we talk about here? Well, we hopefully made the case that the cytoskeleton is an intricate network of protein filaments that extends throughout the entire interior of the cell. The cytoskeleton is not only the bones or the structural component of this cell's interior, but it's also the cell's muscles, allowing for all of this movement. The cytoskeleton is built using three different types of proteins: intermediate filaments that are purely structural for tensile strength, microtubules that are for internal motion of things in the cell, and actin that is for cellular movement itself. So again, the specialty of intermediate filaments is tensile strength, the ability for a cell or tissues of cells to withstand stress without tearing. They're constructed like rope, bundles of fibers that are bundled into bigger fibers that are bundled and braided into bigger fibers, and that gives the tensile strength that intermediate filaments offer. There are four main classes of intermediate filaments: keratin for epithelial cells, vimentin for connective tissue, neurofilaments for neurocells, and then the nuclear lamins that make up the nuclear cortex. And we told a little bit of the mitosis story there for prometaphase. Unlike intermediate filaments, microtubules have a point of origin. All microtubules originate from the centrosome, where there's that gamma tubulin ring. Microtubules themselves are made up of subunits called tubulin, alpha and beta tubulin, that come together to give these dimers. Dimers are stacked up on top of each other end to end to give a protofilament, and then 13 protofilaments side by side make up the hollow tube of the microtubule. Because of this alpha beta dimer configuration, microtubules have polarity. They have a positive and a negative end. And if they didn't have that polarity, the microtubules couldn't do their job of the roads of the cell because the walking proteins wouldn't know what direction they were walking in. Microtubules exhibit dynamic instability for two reasons. One, to make it easy to disassemble and reassemble different microtubule structures for being in or not in mitosis. And two, because the cell is sending out these microtubules blind from the centrosome. And so only microtubules that hit an appropriate target will anchor and stabilize. And within that dynamic instability, please know the GTP cap story, the GTPase on a timer, the instability of GDP containing tubulin dimers, that all falls into dynamic instability. And then we closed out with the walking proteins themselves. N comes before P in the alphabet, and D comes before K in the alphabet. So dyneins walk towards the negative end of microtubules, and kinesins walk towards the positive ends of microtubules. It's these walking proteins that allow vesicles containing cargo to be shipped around the cell and delivered to the appropriate target. And it's also these same walking proteins and the microtubules they walk on that allow the organelles to be placed, stretched, and organized inside the cell the way that they are. So that's it for this video. Again, we'll move right on to actin in the next video and talk all about muscle contraction and voluntary cellular motion. Until then, you know what to do. Let me know what you would like to discuss in class. As always, I thank you so much for watching.